All right, guys, so let's go over some neuropsychology, or it's actually biological psychology. Right here is the communication within the nervous system. Communication within the nervous system. What is the nervous system? What is the nervous system made of? Early scientists believed that the nervous system was a series of tubes. Neuroscientist Santiago Ramon y Cajal identified that the nervous system was composed of distinct cells using various stains. So they thought it was tubes at first, but a neuroscientist named Santiago Ramon y Cajal identified that the nervous system was composed of distinct cells. So they thought it was actually tubes. And I don't know, I guess some sort of liquid or bodily fluid would be sent to the tubes as part of the nervous system. Nissl stain. So there's a Nissl stain, Nissl. So composed of distinct cells using various stains. There's a Nissl stain, only labels cell bodies, and the Golgi stain, a Golgi stain, labels cell bodies and dendrites and axons. So Nissl stain only labels cell bodies, Golgi stain labels cell bodies and dendrites / axons. Also only labels a small percentage of the cells. So the neuron doctrine, they discovered that the nervous system is made up of cells, not tubes. So let's get to the idea of what it is. So main parts of a neuron, you have the dendrites. Dendrites receive signals from other neurons. They're like these little like sticky, like hands. Like the neuron, let's say the neuron's sort of a circle, and you have the dendrites or like the connectors. They look like chicken feet. The dendrites, they receive signals from other neurons. And then other parts of the neuron is a soma, or the cell body, like this. There's a cell body, like the circle is a neuron, and then you have like the dendrites and the axons. So the soma, aka cell body, contains normal cellular stuff, like the nucleus, etc. In this course, the professor said we're not really going to worry too much about the soma. Then we have the axon. So the axon sends signals to other neurons. The dendrites receive the signals from other neurons, and the axons send signals to other neurons. All right, guys, so not all neurons are the same. There's unipolar neurons that contains one axon that receives and sends signals. So let's just paint a picture. So we have one, maybe with only one sort of branch. Unipolar neurons, they contain one axon which receives and sends signals. This is mostly like the lesser developed being, a lesser developed living organism, such as children. Adults don't have, most adults don't have unipolar neurons. And then we have bipolar neurons. It contains one axon and one dendrite. Bipolar neuron contains one axon and one dendrite. And you have multipolar neurons, contains one axon and one dendrite. I think I wrote that twice. So multipolar, that contains one axon and one dendrite. Sounds the same, right? As a bipolar. Other ways to classify neurons. You classify neurons by function. There's sensory neurons, directly receive information from the world around us. Sensory neurons, they directly receive information from the world around us. There's interneuron, which is called the middleman. Then processes incoming sensory information. So the middleman processes incoming sensory information and plans and executes the response. So sensory neurons directly receive information from the world around us. The interneuron, which is also the middleman, processes incoming sensory information and plans and executes a response. Motor neurons connect to muscles and leads to movements and behaviors. So we have sensories, the interneuron, and the motor neurons. Other ways to classify neurons: classify by shape. So one is classified by function, the other is classified by shape. So there is pyramidal neurons, which look like pyramids, and stellate neurons, sort of like stars. Additionally, neurons can be classified based on the neurotransmitter they use. We're going to learn about more of that later. So they classified by function, shape, and also they can be classified by the neurotransmitter they use. So two types of signals in the nervous system. There are electrical signals and chemical signals. Two types of signals in the nervous system. First is electrical. Action potentials signal inside of a neuron. Signal inside of a neuron. Typically used within a neuron. Now the chemical signal of a neuron, which are neurotransmitters, typically used between neurons. So the action potentials, which are the electrical signals, are typically used within a neuron, and the chemical signals, neurotransmitters, typically used between neurons. There will come, getting an idea. We went over this maybe a couple weeks ago. I haven't really done this video, but so there's two types of signals in the nervous system: electrical and chemical. Electrical is the action potentials, usually within the neuron. Then we have the chemical signals, the neurotransmitters, typically happen between the neurons. The electrical signal, the main players to know: ions. So an ion is something that has a charge. The electrical signal, the main players to know. So the main players to know, there are the ions. These are electrical signals, actually, the ions. One of the main ions to know is sodium. So sodium, you remember, an ion is something that has a charge. So one of the main electrical signals is sodium. Sodium is Na plus, so Na positive. It's always positive sodium. Then you have potassium. Potassium is also positive. It's K plus, K positive. Then you have chloride. Chloride is Cl negative. So chloride is negative. So let me try to remember. So sodium is Na positive. Potassium is K positive. Chloride is Cl negative. And then there's some other anions. They're called A minuses, negatively charged in body. There are other anions. For this course, I guess it's not that important. So one thing to keep in mind: negative signals like positive signals. Opposites attract. So two types of signals in neurons are the chemical and the electrical signals. Ways to classify neurons are by function and shape, and also what particular neurotransmitter they use, but we're going to get to that later. Main parts of the neuron are the dendrites, the soma, which is a cell body. So the dendrites receive information, and then the axon sends information. A cell at rest. A cell that is at rest. So cell membrane. A cell membrane. Energy. A cell membrane. Hope your ass went to a spin class. A cell membrane. Um, a cell membrane, a cell at rest. Cell membrane, a fence with gates around the cell. Gates in cell membrane are called channels. A fence with gates around the cell. Gates in the cell are called channels. Resting potential. A voltage difference between the inside and outside of the neuron is negative 70 M volts inside than outside. So a voltage difference between the inside and the outside of the neuron. So let's examine resting potential more closely. Two main forces at work. Concentration gradient. More goes to less. Electrostatic force, which means opposites attract, like changes, repel. Okay, so remember, when we're talking about concentration gradient, more goes to less, like what is more? What more of what goes less? Opposites, what are these opposites? And who's attracting what? Remember, let's go back up to our electrical signals. We have electrical and chemical signals. Let's worry about the electrical signals. Let's go to our ions. Let's worry about the ions. So we have the sodium, Na positive, potassium, K plus positive. We have the chloride, Cl negative, and then other anions, A negatives. We're really not going to focus on them, not worried about them. We just want to worry about, I think that's why he's not using so many different ions. He wants to focus on maybe just the more common basic ones so that we know how the neurons in the nervous system actually operates. So concentration gradient. So I have a chart here, and then we have ions that are inside the cell and ones that are outside the cell. And so when it says more wants to go to less, so say if I'm an electrical signal and I'm mostly inside, and there's not a lot on the outside, there's not a lot of me on the outside, so I want to go where there's less. So I'm going to want to go outside if I'm inside, okay? So that's the concentration gradient. Think about concentration gradient. More wants to go to less. Electrostatic force, opposites attract. If I'm a positive and there's negative on the outside, I'm going to want to go to the outside, right? Just think about that. Just focus on that for now. So the starting arrangement. So the starting arrangement on these specific ions that we're focusing on. Let's just kind of want to worry about the operation of a neuron, the operation of a cell, right? How it operates. We got two electrical signals, there's different shapes, different functions, different neurotransmitters. But there's typically maybe a what do they call it? Unipolar cell, or unipolar neuron, bipolar neurons, and multipolar neurons. Unipolar only has one axon. Bipolar and multipolar contain one axon and one dendrite. We just want to worry about how it operates now. So the starting arrangement. So sodium, which is Na plus, begins on the outside of the neuron. Chloride also begins on the outside. Think about sodium chloride. So one positive, which is the sodium, and one negative, which is the chloride, they begin on the outside of the cell or the neuron, right? So K plus, which is potassium, begins on the inside of the neuron, as well as the anions, which are negative charge. So, oh shoot, a bumblebee. So negative and a positive are inside, and negative positive are on the outside. So sodium and chloride on the outside, one's negative, one positive, and the anions, which are negative, and the positive potassium are inside the cell. So let's think about that. Remember, inside of the cell is negative MV relative to the outside. So let me show you. This is a little diagram. It looks like a cell phone, but think about this as the cell and the outside. Okay? Remember, that's why this is important. So the inside is negative, right? It's considered negative 70 millivolts. I think it might be millivolts. I just say MV, which means if you're looking at this, that's why this is negative, because it's more negative than on the outside. So that's it's like kind of like where it rests. This is like the resting cell position, the positioning of it. And so if you guys could tell, sodium and chloride are on the outside, Na plus, like a bunch of, there's millions of these things. And on the inside, you have potassium and there's, I guess you'll find, they said you find a little bit of chloride in here, not a lot. You got a little bit of sodium. Remember, more wants to go to less. So naturally, these guys are going to want to go inside, and these guys are going to want to go outside. And we'll get to that right now. So this is, and then we have this little diagram here, which I'll read to you. And these positives and negatives will change, guys. These positives and negatives will change. Okay, so right here's a chart. At rest, when a cell is at rest, or is it a neuron? Is it a neuron or cell at rest? A cell at rest. When a cell is at rest, what will each ion want to do? Remember, ion is an electrical. Ions are things with a charge, with an electrical charge. Something that has a charge, an ion. Remember, we know what our ions are. They're potassium, K+, Na+, Ci, negative. We're not worrying about ions anymore. So ions are too large to move, so we won't make predictions about them. So for our class, we don't use A negatives anymore. We're just not worried about it. So remember, we got the ion. It's in columns and rows right here. Columns and rows. We got the ion row, got concentration gradient, electrical force, and then what's actually going to do because of these two things. So K+ is inside, but remember, most of it is inside, so what does it want to do? It wants to go outside. But remember, the overall cell is more negative than the outside of the cell. So since potassium is naturally positive and it's negative inside, right? It's negative inside, but it's naturally a positive ion, has a positive charge. So negative opposites attract, which is the electrical static or electrostatic force. So the electrostatic force is, remember, it wants wherever the majority of the cell is negative or positive, it wants to go to the opposite of what it is. So potassium is a positive, but since the inside of the cell is more negative, it wants to stay where it's at because it likes the opposite charge. So since the concentration gradient, there's more of it inside, it wants to go outside, but because of its desire to stay put, because it lives in a negative charge, it doesn't move. The overall movement is it won't move. It doesn't move. So let's go to sodium, right? So sodium is naturally on the outside, and sodium is a positive charge. It's Na positive. So it's a positive charge. It's on the outside, but it wants to go inside because it wants to go where it's less of it. That's the concentration gradient. But because it's naturally positive, but it's positive outside, it wants to go inside. But it also wanted to go inside because it wants to go where more is less, concentration gradient. It doesn't want to stay outside because it's positive itself and it's already positive outside. So if a concentration gradient is to go inside and the electrostatic force is to go inside, what it's going to do, it's going to move inside. And let's just go over chloride. So chloride is a negative, right? And it at rest, it's on the outside. A cell at rest, chloride is on the outside. And since it's positive on the outside more than it is on the inside, it's going to want to go, remember, more to less, concentration gradient wants to go inside. But it's a negative, and it's negative inside. So it's going to want to stay put because it's a negative charge in a positive arena, let's say. So if it wants to go inside, but its electro force is on the outside, it doesn't move. So right here in this situation, the only thing moving is our sodium. Our sodium wants to go inside. We'll leave it right there, guys, because then we'll talk about what actually is going to happen next. Okay? Which is going to be interesting because we have a lot more diagrams to go over. That's pretty interesting. Peace.